Good morning, everybody. Welcome to Ground Rounds at Jefferson. Today, we have the honor to have a speaker, Dr. Eli Adashi. Dr. Adashi is the former Dean of Medicine and Biological Science at Brown University. She has had a very illustrious career. Dr. Adashi graduated from the Tel Aviv University School of Medicine and followed a residency in obstetrics and gynecology at the Tufts University, and followed by fellowship in reproductive endocrinology and a postdoctoral training in reproductive biology at Johns Hopkins University in Baltimore and UCSD. Dr. Adashi also has a master's degree from Harvard University in Public Health program. Dr. Adashi was the former chairman at the Department of Obstetrics and Gynecology of Utah Health Science Center. She has had multiple awards, multiple grants from the NIH, and he is here today to share with us a fantastic topic for in vitro gametogenesis. Welcome, Dr. Adashi.
Thank you, Amanda. And thank you all for having me. I believe the last time I addressed you was definitely before COVID. Bioslav was still alive and attended the lecture, in which context I wanted to express my condolences to you all on behalf of my wife and I for the passing of Bill, whom we both knew during his fellowship at Johns Hopkins University. At the time, we were living in Baltimore. We were effectively neighbors, and we were in close touch and stayed in touch. Ended up co-authoring some papers, and altogether, my wife and I considered him a close friend. The topic for today is indeed a trend changer. It will change life, professional life, definitely as we knew it. It will probably have some significant implications to in vitro fertilization once it is realized. It will enable what was never possible before, and that is same-sex reproduction. That is to say, to have individuals of the same sex who may actually, with this technology, become the genetic parents of children that would not have been possible otherwise. And so this technology, which, as I will delineate, now dates back to perhaps around 2010, but has been under development ever since, is definitely worth knowing about. And it is probably still a decade or two from being realized, from being approved by the Food and Drug Administration, etc. Nevertheless, it's the future. And for the young members of the audience, it will be probably part of their professional life. The story, you might say, begins around 2009, perhaps even before that, at which time one of the two leading investigators from Japan, Mitinori Saito, published a paper in Cell titled A Signaling Principle for the Specification of the Germ Cell Lineage in Mice. This line of work was expanded by Professor Saito, who is shown here on the left-hand side, and by his close colleague, Katsuhiko Hayashi, who is now working on his own at Kyushu University. But at the beginning, and when I first met the two, they were both at Kyoto University. The experimental paradigms that they have used were predictable. They started with the rodent, and they still mostly focus on the rodent, but are making gradual efforts to make this applicable to the human. Now, all of this really harps back to the notion of pluripotent stem cells. That is to say, cells that could become any cell type in the body. And those, as you probably know, come in two varieties, embryonic or induced, and we will expand on that. But the point is that if you have a pluripotent stem cell, you can guide it to become anything. For example, amacrine neurons in the retina, which are affected in certain disease states. You could potentially make dopaminergic neurons and address the issue of Parkinson's disease in the striatum. Or you can produce motor neurons that could facilitate recovery from paralysis that involves the spinal column. The limit is really one's imagination. But when it comes to gametes, this is probably the leading front of this field, simply because the two individuals I mentioned from Japan have focused on this issue essentially without deviation to a point that has brought us quite far. Ultimately, they seek to produce in vitro derived germ cells that are fertilizable, give rise to a viable embryo that could be transferred and give rise to a pregnancy. When we talk about the so-called gold standards that define in vitro derived germ cells, they obviously have to have the correct nuclear DNA content. They have to have the normal chromosome content and organization. They have to undergo appropriate synapses and recombination. And they need to produce viable euploid offspring. The journal Science, which, as you know, is the leading scientific journal, not just in the United States, understood the significance of this research front, and they dedicated the full spread to mammalian in vitro gametogenesis back in 2021, authored by Professor Saito and Hayashi. The New England Journal of Medicine, not to be outdone, also commissioned an article from Mary Herbert and Azim Surani from the UK titled Oocytes from Stem Cells. Same idea, same topic, but different journal, both of which leading journals that most Americans definitely are exposed to. Most recently, that is last year in April, the National Academy of Medicine convened a 3-day workshop during which the topic of in vitro gametogenesis was carefully and thoroughly delineated as it stood at the time. This will almost certainly be followed by what the National Academy of Medicine refers to as a consensus committee report, that is to say, the product of the deliberations of a committee that can take up to a year, perhaps, of thinking through the subject matter, putting their thoughts in writing, and ultimately publishing a book on the subject, subject to, of course, internal and external reviews. It is instructive to study this infographic, which is produced by the Nuffield Council on Bioethics, which is the leading bioethics organization in the United Kingdom. When they discuss what's on their horizon in terms of what they wish to comment on or study, they make it very clear that topic number one under human reproduction is in vitro derived gametes, the subject matter of today's discussion. Now, they are, of course, going to deal with other issues like time limits on maintaining embryos in culture, womb replacements, that is to say, artificial wombs, preconception screening, whole genome sequencing of embryos and fetuses, genome editing of gametes and embryos, and egg freezing. But interestingly and instructively, at the top of the list, they have in vitro derived gametes or in vitro gametogenesis. We can all agree that this is a disruptive technology that maybe, I put still a question mark here, achievable in the human, although all indications are that this is the case. But the timeline is anybody's guess. I will, of course, show you where we stand today, but like anybody else, I'm not in a position to foresee the future. And so when and if this will become a full-fledged reality remains to be seen. In reflecting on this futuristic topic, it is always helpful to reflect on the work of Aldous Huxley and his 1932 book, Brave New World, which I assume many of you have run into at one point or another in your educational career. In that book, several predictions were made. For example, the notion that eggs will be produced via ex vivo ovarian cultures, that has not quite yet happened. The notion that embryos will be produced via in vitro fertilization, which has very much happened, of course. And the notion that newborns will be created via ex vivo ectogenesis, that is to say, an artificial womb, that also has yet to materialize. But IVF, which was predicted in 1932, definitely materialized ultimately in 1978 in the UK and in 1981 or 82 in the United States. Chapter 1 of Brave New World makes an interesting statement in quotations: one egg, one embryo, one adult normality, making 96 human beings grow where only one grew before progress. That is a statement made by the so-called director of hatcheries and conditioning, who is introduced in the first chapter of Brave New World. Now, this is, of course, not where we are today, although, as you will see, with in vitro gametogenesis, creating 96 wells, each of which contains an egg, a sperm, or an embryo, is very much a reality today. And so, in some respects, Huxley was not all that far from the truth. There are some obstacles to this ambitious goal, and perhaps the most obvious but theoretical obstacle is the so-called soma germline barrier hypothesis, which dominated science quite some time ago. It was formulated by Professor Weissman, professor of zoology at the University of Freiburg, in his book titled The Germplasm, A Theory of Heredity, which was published in 1893. It was Professor Weissman's contention that the germline gives rise to the soma, which we, of course, completely agree with. And if you look at it in that respect, the germline can be thought of as immortal. The soma, on the other hand, never gives rise to the germline, and from that point of view, could be deemed mortal. That notion was turned on its head with in vitro gametogenesis, which is, of course, what we will be discussing today. But all of this is ultimately attributable to the notion of pluripotent stem cell formation and the technologies that stemmed from it. The first such technology was known or became known as somatic cell nuclear transfer, or SCNT for short. The originator of that technology in 1958 was Dr. John Garden from Cambridge University in the UK, who published a leading paper using Xenopus levis as the experimental model in the paper Nature. And specifically what he did is he took a nucleus from the frog, from a somatic cell of the frog, and he injected it into an enucleated oocyte of the frog, a so-called oocytic cytoplast, thereby reconstituting a so-called zygote, artificial embryo, if you will, which developed into a blastocyst. And if you harvested the inner cell mass of that blastocyst, he was able to give rise to embryonic stem cells, which have the capacity to become any cell type in the body, subject to the appropriate guidance by appropriate stimuli that are provided under appropriate timing, which take an embryonic stem cell, effectively an undifferentiated stem cell, essentially in any direction if you know what you're doing. In 2006, the second technology for the production of pluripotent stem cells was introduced, this time by Shinya Yamanaka, MD, PhD, from Kyoto University in Japan, who published a paper in Cell titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. He began with somatic cells from the mouse in this case, and then transfected that cell with select transcription factors, which is the essence of his discovery. And through that maneuver, he was able to take a pluripotent stem cell and convert it into an induced pluripotent stem cells that can now become any cell in the body subject to appropriate stimuli. These two individuals appropriately were rewarded by the 2012 Nobel Prize in Physiology and Medicine, as is shown in this slide. Moving now to in vitro gametogenesis and staying with that topic for the rest of our conversation, we all understand that the story begins with somatic cell, which these two gentlemen we just discussed did in fact do. There is now an established process to convert that somatic cell to either an embryonic or induced pluripotent stem cell, which, as I said now several times, can go anywhere in terms of the cell type we wish to create. And if we wish to focus on gamete, which is, of course, our focus, that is now a theoretical possibility. Now, what are the prerequisites for the accomplishment of this lofty goal? Well, you have to know what amounts to the germ cell specification pathway. The cues that have to be applied to the stem cells, the sequence by which they are applied, the timing of their application, and you also have to take into account the niche or the environment in which those stem cells are enmeshed. And that is because, as you know, oocytes are surrounded by granulosa cells, and sperm are surrounded by Sertoli cells. And those somatic cells constitute the so-called niche, or the home of the gamete, and play an important role in its maturation. And so in the case of germ cell specification, not only do you need to know the cues, the sequence, and the timing thereof, you need to know something about the niche. Now, the beginning of this story, you could say, dates back to 2003, when Hans Scholler, then a postdoc at the University of Pennsylvania, published a paper in Science titled Derivation of Oocytes from Mouse Embryonic Stem Cells, a picture of which is shown on the right lower hand corner. Professor Scholler is now the director of the Max Planck Institute for Molecular Biomedicine in Germany, but at the time was working as a postdoc at the University of Pennsylvania. That same year, Professor Toshiaki Noche from Japan published a paper on the pages of the Proceedings of the National Academy of Science titled Embryonic Stem Cells Can Form Germ Cells in Vitro. A year later, on the pages of Nature, Professor George Daly, who is now the Dean of Harvard Medical School, published a paper titled Derivation of Embryonic Germ Cells and Male Gametes from Embryonic Stem Cells. And that same year, Professor René Rehopera, then at UCSF, published a paper on the pages of Human Molecular Genetics titled Spontaneous Differentiation of Germ Cells from Human Embryonic Stem Cells in Vitro. Over the next 10 to 15 years, multiple papers on the rodent suppress in Nature and Science and Cell, leading journals. And more of the same can be said for the human. Some leading papers, yet again, in very important papers, indicating slow but persistent progress in the field. And so as we look at the state of the art in 2015, we can state that germ cell specification pathway is not conserved between the rodent and the human, which is to say they are distinct, and that the genetic networks involved, as you might expect, are quite distinct as well. What we understood to be the state of the art in 2015 is that there exist embryonic stem cells, be they natural, so to speak, or induced pluripotent stem cells, the development of which transpired in the body in extragonadal tissues, which resulted in their conversion before too long to so-called primordial germ cell-like cells, which are the progenitors of the ultimate mature germ cells that have to be produced within the gonad to yield haploid post-meiotic mature gametes. Back in 2015, we seem to have mastered that piece of the equation, but we yet had to master the intragonadal component thereof. And so the final frontier, definitely in 2015, was in vitro gametogenesis all the way, starting with a primordial germ cell and ending up with a mature gamete. Progress towards that end began in 2016 when Professor Hayashi published a paper in Nature titled Reconstitution in Vitro of the Entire Cycle of the Mouse Female Germline. He started with embryonic stem cell or induced pluripotent stem cells, combined them with primordial germ cell-like cells and somatic cells that allowed primary oocytes to develop, which gave rise to M2 oocytes, photograph of which is shown on the left lower hand corner. Those, upon fertilization, gave rise to an embryo and a blastocyst, which upon transfer to a recipient mother gave rise to pups. In other words, he started with stem cells, he ended with pups. So in the mouse, at least, we know that we can start the process now with stem cells or induced pluripotent stem cells and take it all the way to mature gametes that upon fertilization will give rise to pups. Just in case, because he didn't have somatic cells of mouse origin, Professor Hayashi proceeded to produce those as well using a similar technique. On the male side, more of the same transpired in 2021, this time by Professor Mitinori Saito, who started on the left lower hand side with embryonic stem cell and ended up on the right upper hand side with round spermatid-like cells. In other words, once again, in vitro reconstitution of the whole male germ cell development from mouse pluripotent stem cells, in this case, in the male. Now the question is, will we be able to transmit those insights to the human paradigm? Those efforts are still nascent. In 2018, Professor Saito published a paper in Science titled Generation of Human Oogonia. I say oogonia because we're not talking mature oocytes. Oogonia from induced pluripotent stem cells in vitro. He started with human primordial germ cell-like cells, combined them with some somatic cells, and took the process all the way to oogonia, but not to M2 oocytes. In other words, we're still not in a position to fully reproduce the process of in vitro gametogenesis in the human, in the female. A similar effort was made by Kotaro Sasaki from the University of Pennsylvania on the male side. He was also only partially successful, and in a paper published in Nature Communication in 2020, he reported the reconstitution of prospermatogonia in vitro from human-induced pluripotent stem cell. So again, unable to take the process all the way in the human. So when it comes to human in vitro gametogenesis, we still have to reach the point where we produce mature M2 oocytes and or spermatozoa, both of which are capable of fertilization and the creation, hopefully, of viable embryos. It would also be necessary, best we can tell, to conduct this process in the presence of somatic cells, which in the case of oocytes would involve granulosa and theca cells, and in the context of sperm may involve Sertoli cells. Very recently, in 2023, Professor George Church from Harvard Medical School advanced the process just a little bit by producing functional ovarian granulosa-like cells, which could serve as the somatic cell component for human in vitro gametogenesis. But the likelihood is that much more work will be necessary here before this can be shown to be operational in vitro, let alone is safely applicable in vivo. So what we have now is a scientific and translational promise that may be a decade or two or more away. None of us were, unfortunately, given the ability to foresee the future. And so exactly when and if this will happen remains to be seen. But when it does, there is no question it will have a major impact on the advancement of science. It will provide us with an inexhaustible supply of germ cells. It will allow us to conduct functional so-called null-mutant analysis, which is to say knock out one gene at a time and ask, what does that do? What are the consequences? And thereby learn something about the function of that gene. It would also allow us to study germ and somatic cell interactions, which currently are somewhat limited. On the clinical end of things, it will allow us to, for the first time, reverse germ cell failure. We could embark on autologous gamete replacement in individuals who either lack them or have lost them due to, say, therapy in the course of an malignancy and other circumstances. We could potentially edit gametes in the case of gametes that are genetically in some way malformed and replace those gametes with gene-edited gametes. And of course, this would simplify the notion of mitochondrial replacement, which is a separate topic, but which you may have heard about, the so-called notion of mitochondrial replacement therapy when it comes to mothers who are at risk of transmitting disease that resides in the mitochondria, that is to say, genetic mitochondrial affliction. Now, the likelihood also is that once we can produce gametes in vitro from stem cells, let's say, we may not need to stimulate women with gonadotropic hormones, monitor them with ultrasound, and subject them to egg retrieval, which is a minor surgical procedure. We could potentially substitute all that with in vitro gametogenesis. So in theory, IVG could eliminate the need in stimulation and egg retrieval. It could do away with the issue of gamete donation, which right now is a major element of reproduction. Male donation, female gamete donation is a major issue in multiple circumstances in reproductive medicine that could potentially be done away with. And in a sense, you can imagine a future wherein IVF would, for the most part, be a laboratory procedure, although there will always be the need for endometrial preparation and the transfer of an embryo into the uterus. Will that improve affordability because IVF is notoriously expensive? Will it improve access? Those are open questions. I say so because we have been through this process before and have often been let down. And so I think making open, vague promises is probably not the wisest thing to do. Lastly, it is important to recognize that none of this will ever arrive in the clinic without having been subjected to rigorous examination and regulation by the FDA. This is clearly under FDA jurisdiction. The locale within the FDA wherein this will transpire is known as CBER, or the Center for Biologics Evaluation and Research. And in that particular unit, they will probably refer to in vitro gametes in their abbreviated lingo, HCT/Ps, which stands for human cells, tissues, and products. So going back to where we started, only time will tell whether the Nuffield Council on Bioethics was correct in placing in vitro derived gametes at the top of their list. And I'm making that comment both from a chronologic point of view, but also from an importance point of view, because there are some other important issues in that list. Still, this is a major, major breakthrough if it materializes. And for those of us who grew up on IVF and have witnessed the remarkable evolution of reproductive medicine over the last 50 years, this is probably the next big thing, if it in fact transpires as envisioned. And so it's worth following, it's worth paying attention to, because it's likely going to continue its evolution. The people involved are highly capable and not likely to stop at this point. And my guess is that, particularly for the young members of the audience, this will be a reality at some point, although the precise timeline cannot be established accurately. So the best way perhaps to conclude this presentation is to say, stay tuned. There's more to come. And we will see how all of this shakes out in the next few decades. So thank you very much for your attention and for being there for this somewhat futuristic, but I think ultimately realistic topic. So thank you very much.
Dr. Adashi, this is Vince Berghella, maternal-fetal medicine specialist here at Jefferson. Wonderful to hear your talk, so futuristic. I want to push it even further. From our side, the pregnancy side, this year, CHOP will start having the artificial placenta. They can cannulate, the umbilical cord and probably keep human babies alive from, 18, 20 weeks on. They have already done it in pigs, sheep, and
yes,
monkeys, etc. And they will actually do it in humans this year. On the other hand, on the RAI on your end, we can do amazing things. I was, I guess, in high school or maybe middle school when IVF came around, but lived the almost 50-year story. I guess my question is, how long, we can do all this stuff in vitro, and right now, get to blastocysts, et cetera, but at some point, use a human uterus, quote unquote, still. Do you see a time where that embryo could be kept even longer? Now we have obviously uterine transplants and a lot of the barriers are coming down. I wonder if you think that that's something that scientifically, people are working on.
No, thank you for your question. Although we both have to admit that we are somewhat in the science fiction domain at this point. But I definitely think that, as you know, right now, the convention is not to maintain embryos beyond the day 14 cutoff, although that is being revisited in a variety of contexts, because in non-human embryos, it's become increasingly apparent that we could maintain them in vitro in all likelihood for a longer period of time. I also am cognizant of what you were describing in terms of salvaging premature infants who might otherwise not make it. And if I'm not mistaken, we have either written about it or spoken about it in an earlier point. And of course, in the background to all of this is what you doubtlessly have been exposed to, is the notion of the artificial uterus. And that too is still, you would have to say today, science fiction. But that's not to say that progress is not being made and that interest is lacking. So yes, I think what we could theoretically, and I am emphasizing theoretically, envision is a progressive, quote unquote, replacement of the natural process with an artificial one. Now, I'm not saying this to recommend it, because nature has been refined by millennia of evolution, and an embryo that is transferred at the appropriate time to the womb and allowed to develop in the normal sense to term is, of course, the best way to go, unless we one fine day convince ourselves otherwise. I think those technologies you mentioned and the one I mentioned, to a degree, are backup technologies. They are designed to serve couples and women wherein the natural process has not been fully operational or appropriately operating. So yes, we need to continue to work on this, try to imitate nature to the best of our ability. For those cases where nature does not serve us or cannot serve us for whatever reason. So stay tuned is perhaps still the appropriate conclusion at this time in our evolution, because it's hard to imagine, but no matter what we say, what's coming down the pike is bound to be stunning.
Thank you.
My pleasure.
Good morning, Dr. Adashi. This is Huda Al-Kuwatli, maternal fetal medicine and geneticist. Enjoyed your talk a lot, and certainly we've heard you talk at many other meetings over the many years.
Oh,
Thank you for coming over to Jefferson. So I just want to bring up also one issue, which is the genetic changes that may happen with all of this manipulation that we are doing with the cells. We know IVF in general is associated with increased risk of congenital abnormalities. It is associated with increased risk, for example, of imprinting disorders. And this would be like a separate field, maybe a lifetime field for some individual when this when this research is going to make like actual human progress, as Vincenzo is aspiring, basically. So I just open the minds of our learners, because we have a good number of our trainees who are interested in pursuing genetics fellowships also, in addition to MFM. So thank you for coming today.
no, my pleasure. And I think you and the members of the audience and I are simply at this point, passive witnesses to a scientific development that's occurring in the background. And most people, unless exposed, like you mentioned in previous lectures, or ultimately exposed to the National Academy of Medicine workshop, which obviously gets a lot of hearing and a lot of exposure. But I can tell you, for the longest period of time, very, very, very few people had any notion that this was coming along, because it was all but limited to these two brilliant Japanese scientists who were laboring modestly in Kyoto initially, whom I ended up personally meeting in around 2013 when I was in Japan for another reason. I booked a flight to Kyoto, and I sat down for an hour or two with these two gentlemen and have followed it ever since. But, and I might also add that Kyoto now not only has a Nobel Prize winner in stem cell technology, whom we mentioned, but they have a major stem cell institute. And so there's every reason to believe, given the talents involved, the resources available, that continued development will transpire. Those of you who read Nature magazine, for example, this very week, Nature published weeks, maybe months in advance, a paper by Mitinori Saito, which delineated the epigenetics, not the genetics, the epigenetics of embryonic development, and showed that that can be stimulated and simulated by so-called bone morphogenic protein, a hormone. And Nature, which could see the significance of this work, published it well ahead of when it might otherwise see press, rushed to press, put this out. The senior author, as you can imagine, is Mitinori Saito. So we're talking here a progressive, you may call it evolution, others might call it an explosion of knowledge, but the beat goes on, and new things happen all the time. We just read this paper carefully and have written a brief commentary that is yet being refined before it is submitted, but trying to put it in perspective for the readers. So clearly, you mentioned the young members of the audience who are interested in genetics. They are not going to be bored. Exciting things are underway.
Yeah, absolutely.
Thank you so much for coming and talking to us today.
My distinct pleasure. And hopefully one day we will visit in person.
Congrats. Thank you, Dr. Dashi. Have a great day.
My distinct pleasure. Thank you so much for having me.
Thank you, Dr. Adashi. I will be sending you the recording later today.
Really appreciate it. Thank you very much.